bioinformatics lab interested in genomics, evolution, and machine learning, and these and other things. But this talk will focus on two aspects, or two or three of those aspects. And I'll start. I like analogies to explain what we're doing, and so I'll try to explain what we're doing here. So if you look at this picture, hold on. That's not on my screen.
Can you click that when you got it?
Sorry, just a second. Okay, now. Let's hope that. So if you look at this picture, you recognize some things, and there are some things that you may not recognize. If you've been diving before, then you kind of know what you're looking at. But there's a lot of things that are fuzzy, you're not too sure. And so the point that I'll make during this talk is that it's going to be a lot easier to understand what you're looking at if you see it in motion. And seeing it in motion means letting this process evolve over time. And for some part of this time, you might not learn too much, but at some point you realize now that you see things moving, there are aspects that you understand a lot better. And so we're going to do the same thing here. And I apologize for the people in the room. There's part of my screen that's going to be blocked by this, but I don't know how to get rid of that. So this is a static picture of a DNA sequence, a portion of the human genome. Is that
move your drastic thing and move it up? It doesn't matter.
Yeah, I don't know how to. Okay, on your screen it's okay, right?
Yes.
Okay,
Yes.
all right. So this is a static picture of a portion of the human genome, and we might be interested in understanding what are the different portions of this sequence doing, or if they're doing anything. And that's not an easy exercise because it's static. And maybe if we're good at it, we can recognize certain things, but actually no biologist and no computer programs are terribly good at it. But if I show you not just this sequence, but the corresponding orthologous sequences in a bunch of other species, then you get a feel for the motion, how that sequence has evolved over time. And if you look at this closely, you will recognize certain things. You might see, for example, that the sequence, the portion on the left, seems to be more highly conserved than the sequence, the portion on the right, suggesting that there's maybe something important going on the left. And if you look more closely, you might recognize that if you look column by column in this alignment, there are certain positions that are more conserved than others, and it follows a certain pattern that suggests actually that the portion on the left might be a protein coding region, whereas the portion on the right is not. And so by seeing motion, by seeing how things change over time or don't change, you can get a hint as to what these portions of DNA sequences are doing. Now, in reality, what we're looking at here is not a movie. It's really a bunch of different endings to different movies that split in the past. And thank you. I'm not allowed to drink that, by the way, but that's all right. So, so yes, these are not. It's not different frames of a movie. It's different endings of a movie that started 3.7 billion years ago. If we really wanted a movie, what we would need are knowledge about the ancestral sequences. At every internal node of this phylogenetic tree, we would need to know what was the sequence there, what changed from here to here to there to there to what we have today in our genome or in the chicken genome or others. So we're going to be working towards turning this into a movie and then exploiting that movie. So turning genomic information about modern sequences into something that looks more at an evolutionary perspective, that's the process that we will call the ancestral genome reconstruction process. I'll very briefly explain that, and then I'll talk about how we can exploit that to do interesting things to understand evolution and to understand modern genomes. So the process of ancestral genome reconstruction is a computational problem where we know the genomes of a bunch of species. Those have been sequenced, and there's many, many more today, more mammals and more other vertebrates and so on, whose genomes have been sequenced. So we know those sequences. We know what is the phylogenetic tree that relates these different species. What we don't know are what are the genomes of species that lived in the past. So the internal nodes of this phylogenetic tree. And we want to guess what those sequences were. And in order to make that guess, we need to know what are the different types of evolutionary processes that are at play. Well, there are things like local mutations like substitutions and indels, and there are broader mutations like genome rearrangements and so on. But if we take all of this into consideration, and if we first succeed at aligning these genomes to one another, and then analyze this alignment to figure out, okay, where are the differences between those sequences and how can they be attributed to evolutionary events, then we can predict for each internal node of this tree, what is the most likely sequence that was present there at that time, okay? And that's based on probabilistic models of evolution. And I mean, it's quite tricky. It's actually very tricky to get the right alignment because those sequences are sequences that, for the most part, are not coding regions, are not evolving under much selection. So they're quite divergent. They're not easy to align, but they can still be aligned quite accurately. And so with this process, we can come up with a prediction about what is, let's say, the 3 billion nucleotide genome of the ancestor of every ancestral node in this tree. Okay, and we can actually do that surprisingly accurately. Of course, we don't know exactly how accurately we can do that because we don't have access to those genomes, but we can perform simulations that let us estimate how well we can do this. And there are certain genomes like this genome here at that is called the Boreal Euterion genome. It's this species that lived about 75 million years ago. That is the ancestor of human and of most mammals, except some of the early branching groups like elephants and so on. So we think that this genome, because of its position in the tree, which is kind of in the middle of this radiation period where there's a lot of speciation events that happen, we think that this one we can reconstruct really accurately, to the point where we think that we can get it about 98% right. That means that we're going to predict a sequence of 3 billion nucleotides or so, and we think that about 98% of the nucleotides in there are going to be correct. Okay. I could tell you a lot more about how to do that. It's actually really challenging, but I don't want, that's not the key point for today. So what I want to discuss is why would we be interested in doing that? Okay, it's a fun exercise and maybe you can do it well, maybe less well, but what's the point? Well, the point is certainly not that we're going to take those genomes and try to recreate living organisms for them. And there's lots of reasons why this would not work. The most damning one is that even if we're super accurate in our reconstructions, there will still be a large number of errors in there that will be so damning that no living organism would be able to be created from that. And that's really not the point. The point is that these reconstructed ancestral sequences are going to provide frames of that movie. And by learning how to watch the movie, we're going to be able to infer interesting things about genome evolution and about the function of different portions of our own genome, for example. Maybe that's a good time to pause and see if there are questions. And
I guess I have one that it's more of a what if. So
In the chat, I'll keep an eye on it. Bianca?
yeah,
go ahead, Bianca.
okay. So I was just wondering if sometimes I hear on the news that
Bianca, we don't
piece-
hear you. We hear you
it's-
very poorly.
Okay, hold on, Bianca. It's just that the it's
can
not.
you hear me now?
I'll just for some reason I don't have access to my buttons when.
Yeah, we can put this.
Oh, yeah. Okay.
I can also type my question if it's easier.
Try again.
Hello,
Perfect.
Oh, perfect.
can you hear me now? Okay. So I've heard on the news before that they found, let's say, a piece of an ancient animal in that beautiful transparent stone, for example. And so it, yes.
Well,
And so
that
it preserves
news,
a
of course,
bit of
was,
genetic information.
right.
Have you ever had a chance to compare?
So yeah. So well, first of all, so this is the whole premise of the Jurassic Park movie, and it's not a crazy premise. You can extract DNA from fossils if they've been preserved correctly. That's why we know now what the genome of mammoth is, or of a saber-toothed tiger, or other species that are extinct today, but that lived not so long ago. Now, and that's definitely, and maybe the most notable example is the Neanderthal humans. Okay, so we also have genomes for those. But I'm talking here about species that lived way earlier than this, and to date, there's no way to extract sufficiently intact DNA from fossils that are that ancient to be able to compare. Okay. All right. So I will quickly talk about one interesting application that answers that aims to answer one very fundamental question, which is, where do we come from? In particular, where does our DNA come from? We have 3 billion nucleotides in our genome. Where do they come from? What gave rise to this mass of DNA? And the analogy that I'm going to use, I'm very fond of analogies. I don't know why. The analogy I'm going to use is, do you know the name of your mother? Yes, you probably know the name of your mother. Do you know the name of your grandmother? Let's say your maternal grandmother. Probably yes, you do. Do you know the name of your great-great-grandmother? I don't, and I bet you don't either. But I bet that your grandmother knows what is the name of your great-great-grandmother. My grandmother is dead, unfortunately, but if I managed to recreate her somehow, I could ask her, and then she would be able to tell me what is the name of my great-great-grandmother. Okay, so that's what we're going to do. And we're going to do that in the context of one particular source of new DNA in our genome, probably the most important. It's the study of transposable elements. So transposable elements are pieces of DNA that have acquired somehow the ability to copy paste themselves. So here we have this red portion of DNA is maybe 1,000 nucleotides long, and somehow it has learned, it has acquired the ability of taking advantage of the machinery of the cell in order to make copies of itself. So here we have a second copy, and over time, these copies will propagate more and more. And maybe at some point in the more recent future, there's a new type of transposable element that emerged, the blue ones, and it started propagating as well. And so today's genome is actually made of approximately 50% of DNA that is the result of these insertions of transposable elements. We say 50% because that's what we can recognize, and I'll tell you a little bit more about that in a second. But we actually don't know if it could not be significantly more than that, and that's what I'm going to be going after. Now, one thing that you notice here is that after a transposable element inserts in the genome, it gradually becomes less and less of the color that it started with, meaning it accumulates mutations that makes it over time more and more divergent from the original copy that it was. And so these mutations make it increasingly difficult to recognize in a modern genome every piece of DNA that is a result of a transposable element insertion. If it's the result of a recent insertion like the blue one, then there hasn't been too much time, too much degradation. So those might be easier to recognize. If there's a lot of time that passed, then a lot of mutations have accumulated and it becomes harder to recognize them. Another analogy here, this is a picture of the surface of the moon. And the surface of the moon is constantly bombarded by meteorites, or I don't know if they're called meteorites on the moon, but let's call them meteorites, that create these craters. Some of these craters are really easy to recognize because they're very recent, like the one in the middle here. Okay. Other craters are much older, much more eroded, and they become harder and harder to recognize. So there are some older ones here, and there might even be a big one here. Is this the result of an ancient meteorite, or is this just something that happens to look kind of like a circle and we're not too sure? Well, that's kind of the question that we're going to be addressing. Here's the real situation here. We see those craters that are formed by the insertion of transposable elements. So here we're looking again at the piece of the human genome and how it aligns to all of these other species. And if you see a thin line here in the pig genome, for example, that means that in this genome, there is no DNA corresponding to this piece of DNA of the human genome. And so this pattern of gaps that we see here is the result of this bombardment of insertions of transposable elements. For example, there's a big gap here. That's the result of the insertion of this element here, within which later on another element, transposable element more recently inserted, and so on. And so it's really very much a surface of the moon, but in genomics terms. Now, there are lots of tools out there to look at the genome and identify what are the portions of that genome that look like they may originate from transposable element insertions. The most commonly used is an approach called homology detection. Repeat masker is one of them. And what this approach does is that it starts from a library of known transposable element sequences. There's something called a line L1. That's one particular type of transposable element. We know what the sequence of the original insertions are. And so what we do is that we're going to take that sequence, align it, let's say using BLAST, against our genome, and identify hits, locations in the genome whose similarity to that consensus sequence is so high that it's unlikely that it comes from another source than from the insertion of that transposable element. So we might find these two hits. In reality, you would find 100,000 hits. Okay. And when you find each of these hits, you can also measure the divergence between the sequence observed in the human genome and the original consensus sequence. So this one might be divergent at 10%. That means 10% of the positions have mutated. Okay, and that's the strategy that's used to annotate transposable elements in those genomes. And it yields a picture like this one, and I'll just highlight a couple of examples. There's a particular family of transposable elements called ALUs that has been extremely active in the relatively recent past in terms of human evolution, maybe the last 50 million years, let's say. And we see a very large number of copies of ALUs in the human genome. And those copies, along the x-axis of this graph here, we have the level of divergence of those copies. So those copies are not very divergent. They have a low level of divergence because they got inserted in the genome recently. They have not had a lot of time to accumulate mutations. So that makes them easy to detect. There are other families that have been active over a longer period of time, and so they have a broader divergence profile. And there are other families, like the Mir family, for example, that was only active meaning replicating itself, in the early mammalian evolution. So the only copies that we see today are copies that are called old, meaning that they're highly diverged from their original sequence. Okay. Now, if we look at this graph here, you see that all the curves are dropping around 30% divergence or 35% divergence. That's the result, that's an artifact really of the method that we use to identify them. And that's because when a transposable element copy has acquired more than 35 or so percent of divergence, then we are no longer able to recognize that it aligns to the consensus sequence. And so we just don't detect that. So this drop here is not saying that there were no active transposable elements more than 100 million years ago. It just means that we can't find them. Okay, well, we're going to ask our grandmother. Okay, so what we're going to do is, well, I've explained how we can analyze the transposable elements of the modern human genome. We're going to go back in time, and we're going to use the fact that we can reconstruct that sequence very accurately. And we're going to analyze that sequence using the same tools that we use to analyze the human genome. So we're going to take the human genome, we're going to apply repeat masker to it, and maybe we're going to find a bunch of copies of the L1 element. And then we're going to look at the ancestral sequence that we've reconstructed. And we are going to apply the same approach to identify copies of this L1 family in there. And when we find them, because we have also an alignment between these two genomes, we're going to be able to map those back onto the human genome, which will reveal most of the time that many of the things we knew already from the human genome are going to be recovered again. But we will also find some new copies. And this is showing what happens actually. In blue are the copies that you only find if you look at the human genome. And those would be copies that got inserted more recently than this Boreal Euterion ancestor. In pink are copies that you find using both approaches. So those would be copies that got inserted a little bit before the Boreal Euterion ancestor. So it's found there, but it's also still not decayed too much in humans, so they can still be detected. And in red are the copies that were inserted way far back in time that are not detectable in human anymore, not because they're not there, just because they're diverged to a point where repeat masker will not find them, but through this approach, we do find. And so this red slice here is what we're adding to the annotation of the transposable elements for this family. And arguably, it's a very small slice, so not a very impressive result. And that's because the line L1 family is a family that was active quite recently. There's not a lot of benefits to doing that for this family, but there are a lot of benefits to doing that on families that are older, that were active a long time ago. And this is an example of that. And so using this approach, we can complement or augment the annotation of the human genome by identify portions of the genome that are the result of these insertions. And actually, we can increase quite substantially the amount of the human genome that is assigned a transposable element origin compared to what was known previously. And we can, so for example, we have a new annotation of transposable elements that covers 142 million nucleotides more of the human genome than the previous one. So that's about 10% more. And interestingly, and I'm not going to have time to go into this, but a lot of these ancient transposable element insertions seem to have contributed portions of the genome that today are particularly important, have acquired a function for the host in terms in particular of gene regulation. And Guillaume Bourque has done a lot of work along those lines. So ancient transposable elements were a really interesting major source of new genome regulation. Okay, going to skip this. We can also use this similar approach to discover new types of transposable elements that are not known at all. Nobody suspects them because they're so old that looking at the human genome, we can never even suspect their existence, and we can discover these using the analysis of this ancestral genome as well. So, but I'm going to skip over this. Maybe I can take a quick pause here before switching to the next topic and see if there are any other questions.
Okay, you can put them in the chat if you want.
Yes.
So the phylogenetic trees, is there extra information in the trees that aren't, like, are they exclusively determined by the sequence?
So, okay, so the question is, where do the phylogenetic tree come from? And they come from the sequences, yes. And so it sounds like there's a little bit of circularity here because we take sequences, we infer trees, and from those trees, which we now assume are necessarily correct, then we're going to infer ancestral sequences. But those trees are actually very well established by now if we study mammals. So there's not a lot of uncertainty whether the closest relative to human is a chimp or dolphin. And there's actually a lot of data that supports those trees. So it's not too big of an assumption to make. Okay.
There's a question from Leon. Do the transpositions occur randomly or are they under genetic control?
Right. So good question. So do the insertions of transposable elements happen randomly or under some sort of control? And to a first degree of approximation, they happen randomly. The location in which new transposable elements insert is essentially random, although it might be impacted by things like the openness of chromatin and other things. But to a first degree of approximation, it is random. It's certainly random also in terms of the consequences that these insertions may or may not have. So transposable elements will happily insert inside a gene and destroy the function of that gene. It's just then that the individual who has that insertion may be maybe less fit. And so over time, we will not see mutations or transposable elements inserting in places where they disrupt function. But that's not because there's any mechanism to protect that. It's just a result of natural selection. Okay.
Do different elements have different speeds of decay or change?
So, okay, the CLS question is, do different types of transposable elements decay at different rates? And the answer is, again, to a first degree of approximation, no. They decay at the same rate as, so I call it decay, but it's really just mutations happening. Mutations happen everywhere in the genome, including in those transposable elements. And again, approximately at the same rate everywhere. Okay. Although there are interesting exceptions to that. Okay, I want to talk now about how we can make use of evolutionary information to understand and identify particularly critical regions of our own genome, in particular those that are involved in gene regulation. So a big challenge in genomics these days is understanding gene regulation and how that's encoded in the genome. So you have here an example of a portion of the genome at the top. You have the gene annotation, and by now the accuracy of the gene annotation is pretty good for the human genome. And you also have an attempt at mapping out regulatory regions in the genome. And those efforts are still very much ongoing. And these regulatory regions cover a much larger fraction of the genome than the protein coding genes themselves. So, and mutations, for example, in these regulatory regions are equally detrimental as they would be often inside the genes themselves. So there's a human health aspect to this question as well. I'm not going to go in the great details of how gene expression is regulated. I'll just mention that there's regulation at multiple stages. The first one is transcriptional regulation. So if you want to have a gene produce a protein, you first need to transcribe it, and that's regulated by the promoter of a gene and by other regions called enhancers, where transcription factors, which are proteins, bind and somehow assemble to recruit the RNA polymerase and initiate transcription. There's also regulation to what's called post-transcriptional regulation. So regulation at the level of the transcript that's produced, and that includes things like translational regulation. At what rate are you going to translate a messenger RNA? It also includes things like where is this RNA going to be localized, or how long is it going to stick around? And that is also mediated by proteins that are recognizing certain signals in this sequence, binding to those locations, and somehow impacting some of those regulatory processes. There's been a huge effort worldwide to map and understand these regulatory regions, and there's been great headways that have been made in that direction, but there's the complexity of this mapping effort is staggering, because although every cell in our genome essentially has the same genome, every different type of cells in our genome have different subsets of their regulatory regions that are active. And so we need not only to understand the human genome, but we need to understand it in the context of different cell types. There's thousands of transcription factors and RNA binding proteins that are involved. Our cells can be under different conditions. You might be infected by COVID, and maybe that changes the way in which gene regulation is happening. And in reality, each of our genome is different. And so in principle, we might want to perform this mapping exercise one individual at a time. Multiply all of these things, this becomes entirely impractical to do experimentally. And so there is a need for computational approaches that would have the ability of learning this function that I described here, this function f, that would be a function that's able to take as input a piece of DNA or maybe a piece of RNA and predict what is the activity that this, maybe what is the regulatory activity that this sequence may have. Is it going to be an enhancer in neurons, or is it going to have a role in regulating splicing in I don't know, in stem cells, et cetera, et cetera. Okay? So if we were able to learn a function like this, or a set of functions like that, we would be in a great position, because we would be able to do not only identify regions of the genome that have properties that we want by just scanning with a certain sliding window and applying our function to each of those locations and figure out which portions have which predicted function, but we would also be able to predict what is the impact of mutations by comparing the predicted function of one sequence versus the predicted function of its mutated version. We might be able to understand better the mechanisms that underlie how this function works, the biological mechanisms, and so on and so on. But of course, it's a really challenging function to learn. I'll take a shot at explaining that a little bit more in a second. Just to clarify that we're asking a very general question here. So the type of question that we're going to be asking are, here's a sequence, and here's a certain property that this sequence may or may not have. And the question is, does this sequence have that property? Will it activate transcription in neurons, for example, or something like that? And there's tons and tons of applications of those questions. And there are literally hundreds of bioinformatics and machine learning and biostatistics approaches that have been developed to answer these questions in specific instances of transcriptional regulation or splicing or this and that. Problem is that none of these approaches is working terribly well. And the main reason they're not working terribly well is that they don't have a lot of data to learn from. In order to figure out what that function is, we need to be presented with a lot of examples. And often we don't have access to a lot of examples. Often because in reality, there's not a lot of examples of this happening in cells. So for example, there are certain types of RNA subcellular localization patterns that are specific to really a handful of genes. So if we want to understand that, we're not going to be able to make use of sophisticated, complicated kind of machine learning approaches, for example, because we just have too few examples to learn from. But as maybe you see where I'm going, if we were to use not only information we have about the human genome, but about the entire evolutionary history of this, maybe we would be able to improve the situation. Okay. So the two problems that I'm going to be focusing on here are specific aspects of this, which involve predicting whether a particular protein will interact and bind to a particular DNA sequence or to a particular RNA sequence. And these binding events are really what mediate most of gene regulation. Understanding binding goes a long way towards understanding gene regulation. Okay, and so the kind of question that we will be interested in is, well, here's a collection of sequences that by some means we have determined are bound by a certain protein. Here's a collection of sequences that are not bound by that protein. We want to learn the function that would be able to then look at the sequence and predict whether the sequence belongs to the positive case or the negative case. Okay? And the approaches that we're going to take, which are by now the most popular, are based on neural networks. And so I'll very, very quickly give you a primer on neural networks. Neural networks are regression on steroids, if you want. So their neural network is something that computes a function of certain inputs. Here, the inputs are these 4 red nodes. And the output, the value of the function, is going to be obtained here. Neural networks are typically arranged in layers. And on each layer, you have a collection of nodes. Each of these nodes takes as input every value, or actually each node computes a weighted sum of its input, and the weights are the values assigned to each of those edges. Okay, so the output of this first node is going to be the x1 times w1 plus x2 times w2, and so on. Okay, and yes. Oh, darn. Okay. Thank you. It's very annoying that I don't get to see what.
Yeah.
Okay, I will kill that. Go away. Okay. So by stacking layers of these nodes one on top of the other, we get to approximate very complicated functions. And the training process for neural networks consists of trying to choose which collection of weights would best approximate the known labels of our training data. So training a neural network is a minimization problem that where we're asking, find the collection of weights that makes the predictions of the neural network as similar as possible to the labels that we have for examples for which those labels are known. And this is a challenging thing for many reasons. The main one is a notion of overfitting and generalization. These networks have lots of weights that need to be estimated, often more weights than there are examples to train from. So there's something that needs to be done about that. In a particular case of making predictions about sequences, well, every nucleotide of a sequence, so here I have a sequence ACAG on the left, if this is the sequence on which I want to make a prediction, then I'll need to encode that sequence numerically somehow. And that could be done, for example, by using a one-hot encoding. So encoding A as 1000 and C as 0100 and so on. So then that sequence becomes a sequence of numbers, and the rest follows. Okay. Now there's, like, as I said, lots and lots of work that has been done, and great progress that has been made using, well, until 10 years ago, using statistical approaches, and then using early machine learning approaches, and eventually using mostly machine learning approaches. But the end result, as I said, and that's true also for the RNA binding aspect, the end result is we have things that work pretty well, but the false positive rate remains so high that there's no way that a biologist would ever prefer to use one of these models rather than doing an experiment, if doing the experiment is possible. So here's what we're going to do about that to try to help that situation. And here's another primer, this time primer on genome evolution. So imagine that the picture that I'm showing here is the space of all possible sequences of all possible sizes. Okay, so it's a space of size 4 to the whatever. Okay, so every dot, every black dot in this picture is a different sequence. Here's one, and it's actually an infinitely large space, but a discrete space. Now, this space, let's say we're interested in a particular property of a sequence. Does this sequence activate transcription in neurons? Okay, so then there's a particular subset of that space that I've drawn in green here that includes all of the sequences that have that property, and then there's a subset of sequences that don't have that property. And for now, I'm going to assume that this property is a binary thing. Either you have it or you don't. Okay, so and of course we don't know where the green regions are, but let's just see what happens when these sequences evolve. Well, evolution for a sequence translates to moving around that space, right? Because mutations change the sequence, that means you move around. And so when mutations accumulate, you walk around that space randomly, because those mutations are happening randomly, except that it's a biased random walk in some sense, because if a certain sequence has the property that we're interested in, and if it is important for the organism that this sequence has that property, then it can evolve however it wants as long as it preserves that property. But if a mutation breaks that property, then the individual who has that mutation is disadvantaged. And so that mutation would less commonly be passed on to the next generation, because this individual would have no children or fewer children. Similarly, when a sequence evolves, a sequence that doesn't have that property, if a mutation makes it acquire that property, then that too would generally be detrimental, because you don't want this property to be used randomly in the gene. Okay, so there's a certain selective pressure to either stay within the green region or stay outside of the green region. And so if we had access to the traces of evolutionary traces of a large number of examples, we might see something like that. A lot of these traces are staying within the green region. A lot of the traces are staying within the white region, but there's not a lot of them that are crossing. Well, there might be a few. Once in a while, there will be sequences that acquire a function where it's useful, or there might be cases where a functional region will lose that function. But those are going to be relatively rare. Okay. Now, in reality, evolution is not just a random walk like I was describing it. It's a branching process, and that's what leads to these trees. So in fact, we don't have just these wiggly lines like I was drawing. You have these wiggly trees. If we look at, so this first tree here might correspond to the evolution of one particular portion of the genome, and each of these trees would be depicting the evolution of different portions. But the point is that trees like the one on the top left should be more rare than trees like the one on the right or at the bottom, because this one involves crossing that boundary. Now, if we go one level of refinement further, a lot of functions in biology are not binary, but they're more quantitative. So a certain enhancer might activate the level of expression of a gene to a certain degree. And it might be important for that enhancer to activate this gene at that degree and not too much more and not too much less, because the balance of the cell might depend on that level. And so imagine that the same sequence space that I had here, now look at this as a contour map of this space. There are certain regions where these sequences have a high level of activities. Those would be the green ones. There are regions where the level is very low, and there are regions in between. And so now, if we have sequences that are evolving in this space, they would tend to stick around the contour lines of this map, because it's beneficial, or at least it's beneficial to maintain the level of activity rather than alter it significantly. Okay, so we might have a picture that would look like this. Now, in reality, while we are able to observe individual human sequences and, as we said earlier, recreate their evolutionary traces, we don't know, we don't have the coloring that I was showing before. So we don't know which of those are functional and which are not. But we can assume that this picture here is tracing the contour lines of an unknown function. And we might want to ask, okay, what is this function that has roughly those contour lines? That might be a very difficult thing to answer, but maybe we can do an experiment on every human sequence, or maybe a ChIP-seq experiment was done, for example, that tells us, well, the endpoints of each of those traces, which are portions of the human sequence at different locations, here are their activity levels. Okay, we don't know what the activity levels of the ancestral sequences were, but we know what the activity levels of the endpoints were. And from this, we can hope to infer this entire function with hopefully a decent degree of accuracy. Now, it's getting a little bit late, so I will jump to just one example of doing this. Well, okay, I'll take 1 minute to give you a big picture, and then I'll dive in one specific example. So there are different ways in which we can exploit evolutionary information to improve the accuracy with which we can predict function. The first one is the idea that if we already have a model that is able to look at the sequence and make a prediction, but we don't like that model too much because it's not highly accurate, then we can hope to boost the accuracy of this model by applying it not only to a human sequence, but also to the sequence of the orthologs of that sequence. And by combining the predictions that are made on the human sequence and on the orthologous sequences of other species, we can actually reduce the error rate of the base predictor and improve the overall prediction accuracy. So this is interesting because that's saying that for any type of function that you might be interested in predicting, if you have a predictor that is doing a decent job, but you would like it to make a better job, you can use an approach like this one to use that pre-trained predictor without having to change it at all. You're just going to apply it to every sequence in your alignment, combine the results intelligently, and I'm not going to go into details of this, and get a pretty significant boost in your ability to accurately identify which of those human sequences have the property that you're interested in. Okay. Now, the other approach that I will talk a little bit more about, and which is more related to the picture that I was painting earlier, is this idea of using evolution as a way to regularize training in machine learning. So I'm not going to explain what regularization is for neural networks, but it's a way to basically limit the the capacity of your network, which allows you to train networks that have maybe millions of weights in them, but train them from a smaller number of examples while still hopefully generalize. So it's a way to constrain that. And here we're going to use evolution as a guide to help us train complex models while still avoiding too significant overfitting. And so here's the general idea. In a typical machine learning context, as I explained earlier, we train the model, which means we try to choose the weights in our neural network in such a way that the loss, which is the errors that are made on your training data, in such a way that this loss is as small as possible. Okay. The problem often is that there's lots and lots of different ways to choose these weights that will essentially lead to the same loss. And so there's some guidance that's needed in order to converge on a good choice of weights. And here's where the guidance is going to come from. We call this the phylogenetic inconsistency loss. And the idea is going to be that we will try to favor a neural network that would make predictions on orthologous sequences that are consistent with one another. We would not like to have a predictor that makes a certain prediction on a human sequence, but then it makes the opposite prediction on the chimp sequence, because we expect most of the time that the reality is that those two sequences would have the same function. And so we're going to penalize neural networks whose predictions would be too variable along the branches of this tree. And that's going to push the learning toward a better choice of weights. And hopefully that will translate into more accurate predictions. And I'm not going to bore you in looking at details here, but the bottom line is that it does enable us to improve upon the very best approaches in the field by just, as I said, using this kind of phylogenetic or evolutionary guidance. Okay, I'm going to skip over this last item, except to say that until now, everything I've said in this context makes the assumption that this function that relates DNA sequences to function, biological function, this function does not change over time. That's the assumption that we've made, that we want to learn it, and it's the same in human and in mouse and in chimp. But it's not always true that this function does not change. It is largely true that the way in which a human interprets its DNA is essentially the same as the way a chimp or a mouse interprets its DNA. For example, you can often take a piece of human DNA that might be an enhancer, for example, and put it in the mouse genome, and it will behave in the same way in the mouse genome as it would in the human genome. But it's not always true. Sometimes the mapping between sequences and function changes, and maybe it changes because the environment has changed, or maybe there's some variable that you don't know about that has changed and that has modified this function. And so in some contexts, it's going to be helpful to be able to learn this function, but let it evolve and not be exactly the same in every species. This is what this project is about. If you want to know more about this, I'll be happy to discuss. But I think I will wrap things up here. I will just end by saying that so evolution has been going on for about 4 billion years. It's a super massively parallel experiment where natural selection and mutations generate diversity, and natural selection selects which of those mutations are beneficial or at least not too detrimental. And by sequencing and analyzing the genomes of modern species, we get the readout of this massive experiment. And I feel that we're still not very good at interpreting this, but you can imagine how much value is in this in this data. And now that it's becoming significantly easier to sequence those genomes, and we have hundreds of those, I think there is great value in trying to get better and better at interpreting the evolutionary movie, if you want. I'll stop here. I'll just mention that the work that I presented, the transposable element work, is largely the work of Irina Hiatabibi. The evolutionary work is mostly the work of Faizi Hassan and Elliot Lane. All of those are students in my lab. Thank you, everyone, and I will now take a pause and answer any questions.
Thank you very much, Mathieu, for a wonderful talk. And yes, we are now open for questions. Please either raise your hand or maybe put a note in the chat saying that you have a question. Go right ahead. I'm going to try and keep my eyes on everything, but if I don't, you can also turn your microphone on and say something. Oh, I see somebody.
Jeffrey, you have a question.
Hi, thanks for this great talk. Amazing content. And one thing I was wondering is the transposable element work you've done in terms of looking back in time. Are you thinking of using this to potentially improve the current, I guess, maps of transposable element that exist?
Yeah, so that is what we're doing, and we're about to submit that paper. But yes, it will augment the existing maps quite significantly. Yeah.
Thanks.
Thank you.
Well, I'm going to take the opportunity to ask a question. You mentioned on your last slide environmental control. Is this going to be inferred environmental influence, or is it sort of measured, or?
Right. So no, would this be so these external changes that would alter the way in which sequences translate into function we're not going to attempt to figure out where they come from or what they mean. We're just going to allow this function to slightly be altered, and not entirely. That's the key point, is that we think that, for example, the way human interpret enhancers is pretty much the same as the way mice interpret enhancers, but maybe there's a particular transcription factor in mouse that has changed its affinity, and it's not binding exactly the same way. For the rest, it's mostly the same. So we would like that the human function, the human predictor, is essentially the same as the mouse predictor, except for this small change. And that's what will allow us to learn this family of functions, one for human, one for mouse. But if we had to learn them separately for each species, we would not have enough data to do that. But by insisting that those need to be very similar, but not necessarily identical, we think we can. And in some cases, we've shown that in the particular application of antibiotic resistance prediction in bacteria, it seems to be providing an interesting advantage.
Thank you. Other questions from the floor?
Yes.
You mentioned that like about 50% of our genome is made up of these transposable elements. That's kind of concerning me, because you also mentioned that these transposable elements can copy paste themselves into the parts of our genome that actually help us to live. And so, is what my first question is, like, what is the percentage of active transposable elements? I guess they could just like stop each other from being able to copy themselves. And is this percentage expected to increase with time?
Right.
Until the probability that a healthy birth goes to zero?
Okay, so if I can summarize the question, I'm not sure everybody could hear it. Lucas is worried about his children having insertions of transposable elements at the wrong places. Or basically the question is the activity of transposable elements increasing over time, which based on the explanation I gave you, you would logically conclude that it would. But it doesn't really, in large part because there are controls within the cell to prevent transposition of these elements because they cause trouble. And so cells have a way to repress transposable element transposition. Okay, and there's a war between transposable elements and these control mechanisms, because in order to survive, these transposable elements need to propagate, but the cells don't like that, and so they try to repress them. So over time, these families, so for example, I was talking about one of these very ancient transposable element family called MIR. That's a family that was active, let's say 100 million years ago. It's no longer active because every copy that remained have been mutated in such a way that they have all lost their ability to replicate. Okay, so they're still there, or their remnants are still there, but they're no longer replicating. There are families that are active in the human genome. It's mostly these ALUs and a few other families that are. So if I have a child, his or her genome will include a few transposable element insertions that were not present in my genome. Okay? And sometimes they will be in critical portions of the genome and they will cause diseases. Okay? But it's not an imminent threat, let's say. Although there are species that are under tremendous pressure of transposable elements where 99% of their genome is transposable elements, and that can cause difficulties because in part because it makes their genome so large that it takes forever to replicate and things like that.
One more question for me.
Yeah.
You have, if you're looking at evolutionary arguments, then different families of gene functions are going to behave very differently. So obviously, like if you want to talk about response to infection, that's one sort of kind of evolutionary pressure. If you want to talk about metabolism, it could be a very different evolutionary pressure. And you want to go cognition, then you may not have very good animal models to compare.
That's right.
So how is that going to change the building and the modeling?
Yes, very good question. I don't know if people heard that, but I will.
I think it should be
Okay, so yes, the way in which we use evolutionary data will have or should depend on the type of function we're interested in and how that function could be expected to evolve. If we're looking at things related to the immune system, we know that there is another type of war going on between, let's say, viruses or bacteria and our immune system, which leads to faster evolution on both sides. And that's not the case for other processes, let's say, involved in development or cellular differentiation that are more, that are occurring at a lower pace, let's say. And so the approaches that I described are probably not the best suited for situations like those related to studying the immune system or studying a function that would only exist in the very small or only exist in human, for example. But I would argue that that's still okay because most of the functions that are encoded in our genome are present to a good degree in most other mammals. Not everyone, and maybe in some sense, maybe the most interesting ones are not, but there's still so much that's unknown about functions that are shared across all mammals that it's still a question worth asking.
Thank you.
Good.
Last chance for a question, everybody. Please go ahead.
Right. Well, thank you, everybody. I'm looking forward to hearing more talks later this morning.
Yes, indeed. Thank you again, Mathieu, for a really wonderful talk. It was a great pleasure to have you here today.
Likewise.
To everybody here, we will start back again at 11 o'clock in about 20 minutes. Please go ahead and grab yourself some of the wonderful pastries